page 107 that we'll look at, and hopefully this explanation will make a little bit more sense. Let me zoom out just a little bit. There we go. Alright, so let's look at this in a little bit more detail, what these photosystems are and what this electron transport chain is. Here's this picture. Like I said, it's a good picture. It's found on page 107 in the book. But what's happening is light is being absorbed by the chlorophyll found in the thylakoid. Alright, the chlorophyll is going to store that light energy in electrons. Those electrons are going to pass through the electron transport chain found in the thylakoid membrane. As it does this, it's going to move through structures called photosystems. There are two of them. There's a photosystem 2 and there's a photosystem 1. As these electrons pass through photosystem 2, they're going to pull extra hydrogen ions into the chloroplast. Okay, same thing in photosystem 1. Light's going to be absorbed. These electrons are going to pass through photosystem 1 in the electron transport chain. It's going to pull more hydrogen ions into the chloroplast. Once these excited electrons pass through, the energy from them is going to be used to help make NADPH, which is a type of energy molecule. This excess hydrogen ions that were pulled in through or into the chloroplast through photosystem 2 and photosystem 1, they're going to be pumped through a structure at the end of this electron transport chain called ATP synthase. So this structure is called ATP synthase. It's a type of protein. Okay, and what it's going to do is, as each hydrogen ion is, a new ATP molecule is made. So each time one of those hydrogens move through ATP synthase, another ATP molecule is made. So this is showing how the NADPH molecule, the energy molecule, is going to be made, and how the ATP molecule is going to be made. So it's a very good visual of this process. So here are the words that we looked at on how it works, and then here's a little visual on that electron transport chain, why the chloroplast goes through this light reaction part, and its main purpose is to make, for the light reaction, the main purpose of the light reaction is to make NADPH and ATP. Alright, so that is part 1 of photosynthesis. Let's look at part 2. Part 2 is known as, and I don't like this term, dark reaction, so just forget that term. I don't like that term. This is the light independent reaction. We're going to call it the Calvin cycle. This is the name of the second part of photosynthesis. This is the synthesis, spell it right. This is the synthesis part of photosynthesis. Alright, so this is where something's going to be made. This occurs in the stroma of the chloroplast. So if we look back at our picture, the stroma is that liquid part, the inner liquid part of the chloroplast. This is where the Calvin cycle is going to occur. So this is our Calvin cycle. Alright, it's going to occur in the stroma. Now it can happen with or without light. So it can happen during the daytime, it can happen at night. It doesn't need light to happen for this process to happen. Usually it occurs during the daytime, but it doesn't have to. Alright, so just basic, and then we'll get into the details. So what's going to happen in the Calvin cycle is carbon dioxide is going to be added to a 5-carbon compound. This 5-carbon compound We're going to call it, it's called, fancy word is really long, so we're going to shorten it to RUBP. I might refer to it as Rubisco as well, but it is transferred to this to RUBP. The reason it does this is because it needs to take the carbon out of carbon dioxide and make it usable, because right now carbon dioxide is a gas. So when it takes carbon dioxide in, it's going to add carbon dioxide to an RUBP molecule to separate the carbon out of it and make carbon useful, make it into a useful form. This process where the plant takes carbon from carbon dioxide, combines it with RUBP to make it usable. This process is called carbon fixation. In other words, it means it's fixing carbon into a usable form. When it does this, it's going to form a 6-carbon compound. This 6-carbon compound is then going to split apart. Smaller 3-carbon compounds. The phosphate from ATP and the electrons that NADPH carries are going to be added to these 3-carbon compounds to eventually make glucose. Alright, so in the light reaction part, the first stage of photosynthesis, the end products were ATP and NADPH. ATP is an energy molecule. NADPH is going to carry those excited electrons, if you will, or energy from those excited electrons, and it's going to get transferred, or it's going to get used by the Calvin cycle to eventually make glucose. Alright, so if we go back to our chloroplast, we're going to bring in carbon dioxide. This is going to come in through the stomata of the leaf. It's a gas. It's going to come in through the stomata of the leaf. It's going to be transferred to the chloroplast. And then in the stroma, the Calvin cycle is going to use that carbon, and eventually it's going to make sugar. And this happens in the stroma, the second part of photosynthesis. Alright, so let's get in a little bit more detail about this carbon dioxide, or this Calvin cycle. You have this sheet, and if you don't, you need to get it. Let me zoom out a little bit. Alright. This is a pictorial representation of the Calvin cycle, so it's of this part right here. So in the Calvin cycle, we know carbon dioxide is going to be brought in. Remember, all of the stuff that comes into the chloroplast and leave the chloroplast are going to come into the leaf and out of the leaf through the stomata, those small openings on the bottom of on the leaves. Okay? There are 3 phases, or 3 parts to this. Phase 1 we're going to call carbon fixation. And what is happening in phase 1? Here is this RUBP molecule, alright, that's going to combine the carbon from carbon dioxide, or carbon dioxide is going to combine with this RUBP, this Rubisco molecule, okay? And the reason it does this is to make carbon a usable form. Or it's going to convert the carbon and carbon dioxide when it combines with RUBP into a usable form. The process is carbon fixation. This process is done by the enzyme rubisco. Okay, so the enzyme rubisco is actually what is taking carbon dioxide, combining it with this RUBP molecule, and then turning that carbon into a usable form so the plant can use it, so the chloroplast can use it. That's carbon fixation. That's part one. It takes a little bit of energy for this to happen. Okay. The second, this is the Calvin. Let me go ahead and label that. The second phase of the Calvin cycle is called reduction. Okay, and what this means is that the energy from our NADPH and our ATP from ATP and the energy is used to convert this new molecule. Okay, we're going to call this molecule, it's a biphosphate glycerate, or biphosphoglycerate. We're going to call it PGA. It's going to convert it into a new molecule called G3P. And G3P is this one, this glyceraldehyde 3-phosphate is what G3P stands for. So the energy from NADPH and ATP is going to convert this biphosphoglycerate into this G3P, and the G3P is a very important molecule that's going to be made. Okay, this is the reduction part. After this G3P molecule, these G3P molecules are made. Okay, two of them, so I'm going to say times 2, are going to exit the Calvin cycle. So instead of this 1, I'm going to say 2 right here. It's going to exit this Calvin cycle. So these are the G3P, just a sugar. And this G3P is going to be used to make glucose, which is the big end product of Calvin cycle. And actually it's going to be two, put a little note down here, it's going to be, we'll put an asterisk, 2 molecules of G3P are going to combine to make glucose. And that's the end product this is looking for, is this glucose.